Hello, my name is Julia,
And I'm Jeff.
and this is a brief interview talking about um, Jeff's experience as a researcher and a scientist, and also touching on the connection between Alzheimer's disease and Down syndrome.
Yeah, I hope you like our video today. I'm happy to be here with Julia.
Yeah, thanks for coming on here.
Great.
Thanks for being on here.
I'm happy to speak with you.
Can you introduce yourself and the work you do here at Northwestern?
Yes, I would be happy to. My name is Jeffrey Savas. I'm an assistant professor of neurology at Northwestern University Feinberg School of Medicine. I have a PhD degree in biomedical science, and I lead a biomedical research laboratory where we study preclinical models of neurological diseases, primarily focusing on Alzheimer's disease. In terms of my day-to-day activities, I spend a lot of time reading and writing grants and scientific papers. And I also spend a lot of time mentoring, coaching, and guiding the experiments performed by my lab members.
Cool. Can you describe your lab's mission at present?
I love this question. And the tone and scope of the word mission is right on target because we do have a mission. And our mission is to identify the proteins and proteomes that are responsible for synaptic dysfunction and neurodegeneration as it relates to aging. The idea ultimately is to identify proteins that are dysfunctional and represent drug targets for therapeutic intervention. And the driving philosophy is that if we can identify very early changes, we may be able to modify the trajectory of disease progression.
Can you describe your career path and how you got into medical research, specifically neuroscience and Alzheimer's disease?
About 10 years ago, I became super interested in how mild cognitive impairment potentially leads to neurodegeneration in Alzheimer's disease. And so mild cognitive impairment is common in aging, but not all folks who age develop Alzheimer's disease. And it's particularly relevant because by understanding the relationship between mild cognitive impairment and the transition to Alzheimer's disease, we may have an opportunity to prevent that transition. Alzheimer's disease is very complex, and one of the enigmas of Alzheimer's is that the core pathological hallmarks, the presence of amyloid plaques and neurofibrillary tangles, are required for positive Alzheimer's disease diagnosis, but they are not sufficient. So the goal is to identify patients and or studying models by which we can have model systems to investigate and probe the earliest stages of the disease and potentially prevent that transition. And as a child, I was very interested in biology and chemistry. I had like every type of pet you can imagine in a chemistry set in my basement. And as I went to high school and college, I became very interested in cells and molecules, and in particular, biological macromolecules. And proteins are among the most important biological macromolecules, and they're incredibly diverse, and they have a remarkable diversity in terms of their structure function. And they play a key role in functioning as the micro machines inside of our cells. They're actually the effector molecules. They're the molecules that have a specific job. And that my interest during undergrad grew specifically during graduate school, where I focused studying proteins, and I became aware about a class of diseases called proteinopathies. And proteinopathies are a class of neurological disorders that seem to manifest at the protein level. And what is common to all proteinopathies is that the patient brains from people with proteinopathies have protein accumulations. And I specifically studied Huntington's disease, which is related to Alzheimer's disease, but slightly distinct. So then, after I completed my PhD studying Huntington's disease, I then went and did my postdoctoral research to master and increase my knowledge of tandem mass spectrometry, which is the analytical technique that we use to study proteins in complex mixtures. And mass spectrometry represents among the most powerful analytical tools to identify and quantitate proteins.
So then how did you get from studying Huntington's disease to Alzheimer's disease?
Huntington's disease is very interesting.
Mm-hmm.
And in theory, it should be a neurological disease, which would be very straightforward to tackle because it's a single gene mutation in the Huntington gene. And we know the mutation, it's a CAG, trinucleotide repeat expansion, which causes for the production of a poly-Q expanded protein. However, Huntington's disease is quite rare, something around the order of 1 in 30,000 people.
Oh, wow.
Alzheimer's disease, on the other hand, affects far more people, something around the order of like 1 in 9 individuals. Mm-hmm. So I thought, in terms of maximizing impact, I thought shifting towards a more commonly observed disease of Alzheimer's disease was a good idea. However, in retrospect, Alzheimer's disease is far more complicated than Huntington's disease. So it's an important problem, but it's also a challenging problem.
Yeah. Along your journey, how did you come to find out about the connection between Alzheimer's disease and Down syndrome?
Another great question. So in thinking about this, my lens has been primarily through a scientific lens. So in my interests in the lab. So about 7 years ago, I spoke to a clinical neuropathologist from University of Pennsylvania. And in a conversation very much like this, we were speaking about my interest, and he strongly suggested that I examine Down syndrome patients or Down syndrome patient brains, because these patients provide a very rare and precious opportunity to study people or tissue samples from people who inevitably will develop Alzheimer's disease.
Yeah.
Specifically, virtually all individuals with Down syndrome will develop the neuropathological hallmarks of Alzheimer's disease by the time they're 40 years of age. Yeah. So this is relevant because if we can obtain brain tissue from Down syndrome patients in their 20s or 30s, we can actually have a chance to view or look at the very earliest transition by which the human brain begins to start to manifest these very initial changes towards the development of Alzheimer's disease. The challenge, though, is that these patient brains are very precious and very challenging to obtain because these patients have to pass away through other means. So typically through some unexpected accident or another disease unrelated to Alzheimer's. And there are some tissue repositories in the United States, but even across the whole United States, we've only obtained as many as 10 or 20 Down syndrome patient brains in their 20s or 30s.
Wow. So can you describe the connection for our viewers in layman's terms? So just very simply, like what it is.
So Down syndrome is caused by the triplication of chromosome 21. So in addition to the neurodevelopmental challenges that these individuals face, they inevitably develop Alzheimer's disease because the amyloid precursor protein, which causes the production and accumulation of amyloid beta peptides, which play a key role in the early stages of Alzheimer's disease, inevitably accumulate in those brains due to overexpression. It's a slightly different situation from sporadic Alzheimer's disease, which actually manifests due to an inability to clear those toxic polypeptides. The Down syndrome patients actually accumulated due to overexpression because of the extra copy of the gene.
What was your reaction when you first learned about the connection?
I was excited by the opportunity to learn about this precious opportunity in human patients. So I was immediately proactive in trying to obtain them.
Yeah.
And that however, took many, many months to actually get in contact with the clinical resources by which the tissues are stored and obtaining tissues that were preserved in the right way. But it was an exciting moment to have a eureka opportunity to get at this question.
What would you say are some of the biggest challenges in doing research?
I personally think the biggest challenge to doing research is being able to multitask. So in an average day of a biomedical scientist, we're forced to be able to perform multiple tasks at the same time. So we're writing grants and writing scientific papers while we're managing our students and meeting deadlines all along the way. And the job never really ends. So it's not a job that has a quitting time. I go home, but I bring my work with me, and I like to read science at night, and it's an all-encompassing profession,
Yeah.
which is fantastic in some ways, but also can be challenging and exhausting in others.
Nice.
Another challenge to being a scientist is the constant fight to publish papers and obtain funding. So the analogy that I really like is a lab is similar to a big wooden barrel that's full of water, and you have holes in the bottom of the barrel, and the water is always leaking out, which is like the money going through the lab, and we always have to find new ways to add water to fill that bucket back up.
Oh, I've never heard that before. And I guess you talked about like some of the challenges in specifically researching Alzheimer's disease. So, I mean, do you want to, I guess, elaborate on that a little bit more?
Sure. So we talked about the challenge in studying Alzheimer's disease in humans because not all patients who have mild cognitive impairment develop Alzheimer's disease. So that's a major challenge. So we cannot predict those folks who will get Alzheimer's, and it can only be confirmed post-mortem by the presence of plaques and tangles and dementia. So then we're back to studying models. And you can study mice, which my lab does a lot of. And so mouse models of Alzheimer's disease can recapitulate some aspects of Alzheimer's disease, but no single mouse model fully recapitulates the complex mechanisms by which Alzheimer's disease manifests in the human brain. This is for a number of technical reasons. Mice don't succumb to neurodegeneration, so they don't lose neurons unless it's an artificial element of the model. The other major limitation is that very few, if any, Alzheimer's mouse models actually develop both amyloid plaques and tangles. Also, obviously, mice only live a few years and the humans live for decades. So the rate by which things change and how the pathology is related to aging is hard to study in mice. Human-induced pluripotent stem cells is another emerging model for Alzheimer's disease. My lab also uses some iPSC-based models of Alzheimer's disease, and the benefit of those models are that they are human. However, these are cells grown on plastic petri dishes, which do not recapitulate the endogenous 3D environment of the brain. And then subsequently, we also work with postmortem tissue, brain, serum, liquid material from patients. But again, that's actually like separated from the human, and that just provides snapshots of the molecules or analytes that are present in the sample at that one time.
Got it. Okay, so we talked about the biggest challenges. What are some of the biggest rewards of doing research?
The most rewarding aspect of being a scientist is watching your trainees grow as scientists and to achieve their career goals. And this, over a typical PhD, is 5 to 6 years, and it's just a remarkable transformation to see someone grow and increase their confidence and really blossom as a young professional. I also am particularly driven by making discoveries, and I sometimes feel as if I'm an archaeologist looking for buried treasure or peering into an ancient tomb to actually see something which is never seen before or has never been observed in that way. This is particularly exciting to me because it's breaking new ground and truly elucidating, or at least envisioning, aspects of disease that no one has ever seen before. So I find a lot of satisfaction there. And I think the final element, I find a lot of satisfaction in contributing to the general knowledge base. So when we publish a paper, it goes to a public repository called PubMed, and that information and those findings will be accessible through electronic records for centuries to come. And it's very difficult to predict the value of a discovery at any one time. So in this way, future scientists of today and tomorrow can actually go back into the literature and see what we found and then try to use that information to accelerate their own studies, which is an exciting possibility.
Yeah, for sure. That is very exciting. Okay, finally, do you have any words of encouragement or advice for young aspiring scientists?
I do.
First suggestion is to identify what you're interested in. And the way that interest in science goes is we are only able um, to learn or focus on things that we're exposed to. So by exposing yourself to many different topics, you allow yourself an opportunity to home in on specific areas or aspects which you may find interesting. So that's the first step. Um, I think the next step is that as a scientist, you have to be process driven and understand that it's going to take a lot longer than you may realize. So, Julia, I think as you know in the lab, it takes a long time to do experiments, and a couple of weeks or a few months is really just a short amount of time, and it takes years. But if you work hard and you're passionate about what you're doing and you're creative and you're willing to go on the journey, I think that you're destined to find exciting things and make important contributions.
Thanks so much for watching, and thank you for coming on here again.
It was a pleasure to speak with you, Julia.
We hope you learned something new through this video.
Thanks for watching.
Thanks for watching.